Hey everyone, welcome to another episode of the Takeaway Club, where I talk to interesting people across different walks of life to get to know their stories and, more importantly, how they got to where they are, including their motivations, habits, routines, and aspirations, some of which we can apply to our own lives. Hey, before we get into the episode, a quick update. Along with the podcast, we now have a newsletter. It's called Beat the Blues, a bite sized curation every Monday with the most interesting and random content that I come across in the week prior. Earlier, I used to do this in an ad hoc manner, sharing an article, giving book recommendations to my friends. That's when one of my friends suggested why not do this in a more organized fashion, and that's why we turn this into a weekly newsletter. It's a small non-intrusive email every Monday with a bulletin of whatever content I am currently into. It could be the books I'm reading, the podcasts I'm learning from, tweet threads that I enjoyed, just a small boost to kickstart your week, and hence the name Beat the Blues. So if you're someone who enjoys insightful random content but don't have the time to sit through and do the dirty work of browsing to find them, you're going to love this. It's super easy. Just head over to thetakeaway.club and hit the subscribe button, and you'll get the very next one. Hey everyone, thanks for joining me on the Takeaway Club. In today's episode, I have Shaswath Sekar, a virologist in training. He's currently pursuing his doctorate from the University of Wisconsin-Madison on veterinary pathobiology. His work focuses on the development of vaccines against animal and human coronaviruses. So without further ado, let's jump right into the conversation. Hi Shashwath.
Hey Krishna, thank you so much for having me here. I'm looking forward to our conversation.
Exactly, man. I'm very excited about this conversation and look forward to learning a lot about your work as well as the pandemic that has completely flipped our lives around.
Yep, definitely. Yeah.
I want to start off with some context on your background, for people listening.
Okay.
So, as a virologist, what does your day look like? So, if you can maybe give a walkthrough of some of the activities that you are involved in a typical work week, that would be great.
So, as a virologist, and this is true for many people in the field of STEM, there is no specific schedule that we follow. A lot of what we do over a week or over a day depends on kind of the projects that we're working on. And the experiments that are part of this project, right? And in general, virologists, there are, they're very diverse, right? The kind of work that, say, a coronavirologist like me would do would be something very different from what, say, a person working on measles would do, right? So I can give you some, perspective from the personal side, right? So my kind of work is focused on vaccine development, and there are two basic aspects to it. One is to develop the vaccine itself, and the other aspect is to test it in the host. So in terms of vaccine development, a lot of what I do, is cloning, right? It's a fancy term that's been thrown around, but it's a very common thing for general bioscientists and molecular biologists. So what I do is kind of chop genes, viral genes from the viral genome, and then insert it on another strand or piece of DNA. And this piece of DNA is something we call a plasmid DNA or a vector. And this vector will help kind of the viral gene enter the target host cell, right? So this is kind of one aspect where you have the vaccine development per se, and then we run a couple of quality control tests to make sure that the vaccine is working as it should before we kind of inject it to the animals. And then the other aspect of it is, of course, testing it in animals. So before we kind of test the efficacy of the vaccine in animals, we make sure that the vaccine is well tolerated by the hosts. there is no allergic reaction, and then we go about testing it. So, for example, with chicken coronaviruses is something that I work on. We vaccinate birds at a very young age, and then we challenge these birds with the actual virus a couple of weeks later, and see if these birds are protected against challenge with different metrics. So a lot of work, some few days I'd just be spending time doing animal husbandry, making sure that the birds are healthy, eating properly, drinking water. And something interesting here is that young birds, chicks, as soon as they hatch, they do not know how to drink water or where to drink water from. So I spend like 2 to 3 hours as soon as they hatch teaching them how to drink water. Forcing their beak onto these automatic water nipples and making sure that they drink water and that they know where kind of the water nipple is. And then I wait for an hour to make sure that the chicks are going there and drinking water from that. So it's just very diverse. So I do a lot of things, and a lot of what I do is kind of dictated by which part of the project I'm in currently. And now I'm working with SARS CoV-2, right? So there is a lot that goes into that as well, because with SARS CoV-2, considering the circumstances and the fact that we don't have vaccines, the fact that we don't have any drugs, it's a very dangerous pathogen, right? So there needs to be very special conditions to work with it. So it is classified as a biosafety level 3 organism. So you need a BSL-3 lab to work with SARS CoV-2. And this would mean that the whole room is under negative pressure and would have HEPA filters, right? These are filters which can filter out the air that's going out and preventing escape of the SARS-CoV-2 that we work with in the lab. And it takes about half an hour just to put on all the safety equipment necessary before we enter the room. This would involve, wearing your scrubs. On top of that, we wear a special suit. You've seen all of these images online and doctors wearing these suits. That's what we do as well. And outside of that, we wear something called a PAPR unit. So this is like a huge hood, like an astronaut that you kind of place over your head, and then connect to a device which is on your waist, which would filter all the air that you breathe, right? So these are kind of the safety precautions that we take before we even start working with the virus in our lab. And then before coming out, you completely decontaminate the suit with alcohol and then bleach. And then once you get out, after you remove all these suits, you need to take a bath to make sure that you got rid of any trace of the virus on your body.
Wow.
And then that's when you can really step out of the BSL-3 facility, So this is a lot of what goes into my work as well. I spend like half an hour trying to get into the lab when I have to work with this virus, and then another 45 minutes to get out of the lab, So it's pretty interesting. It's a lot of fun, to be honest.
It's like throwing up a stormtrooper every time you have to step in and out of the lab.
Yep, exactly.
How are things like at present in terms of the pandemic? Maybe if you can throw some light into the specific kind of work that you are doing with respect to this fighting against this coronavirus. What were the first few weeks like when this thing got blown up?
To kind of give your listeners some context, I joined my PhD initially, and the work that I focused on was to develop vaccines against chicken coronavirus. And these, chicken coronavirus, as the name suggests, infects chicken, and it's of great economic importance in the poultry industry. Just like SARS-CoV-2 and COVID, it causes severe respiratory symptoms. But, the chicken can easily clear the infection within 10 days. But then what happens is, the life of a meat chicken or a broiler is only 9 weeks, 8 to 9 weeks. And then if, they are sick for a week or 10 days, they don't gain as much weight. And unfortunately, the poultry industry is an industry of small margins. So this is something that farmers cannot afford, right? The kind of decrease in weight gain, which is kind of major outcome of avian coronavirus infections. So there is a need for a cure. So the cure that I was working on was a vaccine, right? So we kind of worked on developing a vaccine that could overcome some of the limitations of current vaccines that are being already used. Okay. So once kind of the pandemic started coming up, so this was, I think, late December and early January, since I work with coronaviruses, and it kind of fell on our radar much before it did for the rest of the public. So my boss and I were kind of following it for a while, and then once it started becoming big in mid-Jan, we decided that why don't we try translating this technology for human coronavirus, SARS-CoV-2. And the other thing that I want to note here is that in our lab, we regularly work with tuberculosis, which is even more transmissible than SARS-CoV-2. So we have to take more precautions working with TB than we do with SARS-CoV-2. So we have the necessary expertise to work with a deadly respiratory pathogen, right? So combining that and combining our expertise in coronaviruses in general, we started working on vaccine development for SARS-CoV-2. So at that point, I think we started making our vaccines late January, and we had a vaccine construct which was ready by late February. And then we started testing these vaccines mid-March in mice, and the studies are still going on. We have some good, encouraging preliminary results, right? So outside of this, how has this impacted the general bioscience community? In general, the academic environment in the US, and this is true for other countries as well, is very structured, right? So in a lab, you have a principal investigator or a PI who is essentially the boss or the equivalent of a manager in a corporate institution, right? And he is in charge. He's kind of the think tank. He or she, they are the think tank who come up with the ideas. And outside of that, they are also responsible for getting the money to do these projects. So the money again comes from federal grants, so grants from the government or other non-profit organizations, for example. But they're very specific. So if you get money, say if you get $500,000 to do a particular project, it should be only to kind of perform experiments related to that project. Right? So it's very tough to kind of deviate outside of the project goals, right? So immediately as soon as this pandemic hit, luckily we were in the field of coronaviruses, so we were able to convince people who are funding us that we can translate our expertise for the greater good of the public, right? So, but this is not true of many labs in the STEM field, right? They are obviously 99.99% of the labs are not working on coronaviruses. So they were not able to kind of transition to working with coronaviruses. And unfortunately, many of them had to shut down during the lockdown, right? But then despite this, many labs, they have taken the risk and diverted funds they've been using for other projects to work on SARS-CoV-2, to understand and come up with interventions to help public. And it's just it's like it's the kind of pace that we are in is like a war-like footing. Scientists are racing against time to kind of come up with interventions, vaccines, and therapeutics for SARS-CoV-2.
So, as someone who has been working on this much before the rest of the world,
Uh-huh.
what was your initial response like when you saw this exploding the way that it has now? So, what were
Yeah.
some of the thoughts going through your mind, say sometime in late December or January?
Yeah, so with respect to coronavirologists in general, we've always been kind of outside of center stage. There are other big viruses such as flu and Ebola and Zika. These virologists always get their due, and rightfully, right? These are more important, or were more important than December of 2019. But then coronaviruses have been circulating in animals and humans for a very long time. So for chicken, coronaviruses has been, I think it was recorded, first time it was recorded, I think it was early as 1930, right? And we still don't have good vaccines, right? And that is because, it's in the poultry industry and not humans. And before SARS-1 hit in 2003, coronavirologists, no one really worked on, especially human coronaviruses. There were like four human coronaviruses that cause common cold, right? So there's no real attention. And then once SARS-1 hit, there was more importance given to coronaviruses in general. There were more groups who started working on it. And then, SARS-1 eventually kind of died down. So no one really cared after that. And then MERS hit in 2012 was it? And then again, there was, a spurt in attention. but then it eventually died down because MERS never really picked up. But this one has been insane. This is like a great upgrade on SARS-1 in terms of transmissibility, so on and so forth. But working with chicken coronaviruses, we've seen this with chickens. This has happened with chickens 100 years ago, right? Something similar to SARS-1 first came in the 1930s, right? And every 5 years there were new different variants of avian coronavirus that started propping up. And that is what you're seeing with human coronaviruses too, right? In early 2000s, you had SARS-1. And then early part of this decade, you had MERS. And now you have SARS-2. So we were like, we've seen this before in chickens, right? And people shouldn't take this lightly. Coronaviruses are highly transmissible because for chicken coronaviruses, if one bird in a flock of 10,000 gets infected, within next 2 to 3 days, all of the 10,000 birds get infected, right? So that's how quick it is, right? So we were kind of, we were hoping that it'll kind of die out like SARS-1. But, as the weeks went ahead, we kind of felt like the number of cases were precipitously increasing. So that's when, we started, we were like, maybe we should give more importance to it. And let's start thinking of, developing a vaccine for this,
Mhm. Multiple terms that are used interchangeably when talking
Mm-hmm.
about the infection. Uh, you
Mm-hmm.
mentioned SARS CoV-2 and we call it COVID-19 and coronavirus. So, what is
Mm-hmm.
the distinction between one another? My vague understanding is
Yes.
coronavirus is more
Mm-hmm.
like the family, and then
Mm-hmm.
SARS CoV-2 and COVID-19. It's all, I don't know, something like part of the family along with MERS, SARS CoV-1. Is that right? Or
Yeah, so in a sense, yeah. So as a person working in the field, it kind of irks me that when the media uses these terms interchangeably, but they are completely different. So to begin with, coronavirus is a family of viruses. Okay, and there are 46 different types of species within coronavirus. Okay, so one of those species is SARS-CoV-2, and
okay.
another is SARS-CoV-1, and another is MERS-CoV. And the virus that I work with is infectious bronchitis virus, which is kind of the chicken coronavirus, is also a species of coronavirus. So coronavirus is the family, and it has 46 different type species within the family, right? And one of those viruses is SARS-CoV-2. So SARS-CoV-2 here is the virus. So it is severe acute respiratory syndrome coronavirus 2, because there has already been version 1 in early 2000s, right? And then COVID is the disease that is caused by SARS-CoV-2, right? It's coronavirus infectious disease, right? And 19, obviously, because of the year that it came up in. So it is the same difference between HIV and AIDS, right? AIDS is the disease or the syndrome which is caused by HIV. Similarly, COVID-19 is the disease that is caused by SARS-CoV-2, which is part of the coronavirus family.
So, in a sense, there is a chance that this very SARS CoV-2 might cause something other than COVID-19.
No, COVID-19 is just a broad definition for all of the symptoms that you see, right? And viruses are very specific in terms of the symptoms they cause. So SARS-CoV-2 can only cause COVID-19 so far,
Okay. So,
right? That's all
basically,
we know.
every virus gets one disease that it can infect people with.
Yes, yeah, but we don't know what SARS-CoV-3 or 4 will hold.
Okay. Okay.
What will happen with those. So
Wow. So could you shed some light on the evolution of the virus, since we talk about the family and its neighbors and everyone? I mean, you mentioned there were like
right,
46 types of viruses. We are only what, two down now, right? Or if we take MERS,
3.
SARS, three down. So that looks like there is still a lot where it came from. I mean, I understand
Yep.
it originate this particular virus SARS-CoV-2. Uh, it originated from the wet markets in Wuhan. But
Mm-hmm.
why now? How does this whole thing blow up like this? Is it how does it work in a scientific perspective? Is it a case of there are all these different actions, action A plus B plus C, and then boom, you just trigger a new virus?
Yep. So So I want to give again some context. I always like to, tell a decent story, right? So I'm again going to go on a tangent, but this will be, hopefully will be informative. But in general, what you've been noticing for all of these new emerging infections, right? Take Zika, take Ebola, MERS, SARS-1, and even SARS-2, they have all been spillover incidents, right? So these are all cases of zoonotic infections, right? So this term would mean kind of jumping, the virus is literally jumping from an animal to humans, right? And for SARS-1, MERS, and scientists speculate for SARS CoV-2 as well, that this jump would have come from bats, right? Bats, as you would know, are like the most diverse species when it comes to mammals. I think it's bats and rodents. So proportionally, they have a lot of viruses in them, right? Because of which you see a lot of these jumps from bats to humans. So, and this is for SARS-1, MERS, and SARS-2, right? And outside of these three important coronaviruses, there are other four other human coronaviruses that cause common cold. So 30% of the common cold infections in the world are caused by human coronaviruses, which are not SARS or MERS. Okay, and these coronaviruses, like thousands of years ago, would have been like SARS-1 or SARS-2, right? At some point, they would have jumped from bats to, or some other reservoir. We don't know which animals were the reservoir. Would have jumped from bats to, or that animal reservoir to humans, and now they have become human viruses, right? And that's what's happening right now with SARS-1 and 2. Okay, and with respect to specific things that need to happen for a virus to jump from one host to another, is that viruses are very specific in terms of the host they infect, and also the kind of cells they infect within a host. Okay, and that can dictate what kind of clinical signs host will kind of represent. So for SARS-CoV-2, it uses the receptor called human ACE2, right? So for this kind of bat virus to transition from bats to humans, the virus really needs to learn how to use the human receptor, right? It's kind of a door, and it needs to know how to open this door to enter the cell. So that is one big adaptation, right? And that is one of the biggest barriers that a virus has. So how this could have happened is, scientists speculate that it was not a direct jump from bats to humans, and that there was some intermediary reservoir. Okay, and coronaviruses in general are very prone to recombination. That is, they tend to mutate a lot and tend to recombine with closely related coronaviruses, right? So imagine that there is a bad coronavirus, and then there is in China, for example, say there are pangolin coronaviruses, right? Say, for example, a bat hunter or a wildlife hunter, hunts a bat and then hunts a pangolin, right? So at that point, what happens is there is a chance for bat coronavirus to recombine with this pangolin coronavirus to, develop a new hybrid coronavirus, right? And there is a high chance that this hybrid coronavirus has the ability to use the human receptor, right? And then eventually in these wet markets, when there is just poor hygiene conditions, and then these exotic animals are caged in close quarters, and there is fecal transmission from one animal to another. So there are so many different animal species. Proportionally, there are so many different animal coronaviruses, and then you have humans in this scene as well. So what happens is, humans, we have really put ourselves in circumstances where we are exposing ourselves to these recombinant coronaviruses, and then this coronavirus just happened to like where it came, so it just evolutionarily adapted to humans really well. So I guess the biggest adaptation that this bad coronavirus would have undergone is to learn how to use the receptor for entry, which is the human ACE2, right?
After what you explained, in retrospect, it makes sense why this happened somewhere in the wet market like that.
Exactly. So I think going forward, the ways in which you can prevent such pandemics is to step up surveillance, right? Pump in research to understand what are the different types of viruses that are circulating in these animals, especially bats and rodents, because these are the most diverse mammal species out there, and proportionally, they have a lot more viruses circulating, right? So, if we understand the type of viruses that are circulating and kind of see how far they are from
When you
getting
say
used to,
diverse, I have a
I'm sorry.
I just had this question there. When you say bats and rodents are diverse,
Uh-huh. Uh-huh.
what exactly do you mean by that?
So there are multiple species within bats. There are different type of bats out there, right? And each of these types of bats will harbor different type of viruses, right? Because they have different habitats, different, sleeping schedules, for example. They eat different things. So all of this will also dictate what type of viruses they harbor, because the environment they will be surviving in is completely different, because of which the viruses that they have will also be completely different, right? So, each species of bats will have their own niche environment of virome that they will have, which could, spill over into humans. And does it make sense?
Yeah, got that part.
Yep.
Is there a reason why most of the viruses spread to humans from bats? Or
Yeah, I know.
is there any
Yeah,
correlation
so this
there?
is something that scientists have been trying to figure out, right? So these bats have so many viruses that are circulating. A single bat could have like, I'm just, taking numbers out of the air, but I could be wrong, but there'll be 30 to 40 viruses circulating in a bat at the same time, right? But the immune system of bats are so fine tuned that they're able to keep these viruses at bay. And this is still, the research is still ongoing. No one really has an answer as to why bats are able to harbor so many viruses and are also kind of able to keep so many viruses at bay. But one speculative reason is that, they are flying mammals, right? So because of which, when they tend to fly, they have a higher body temperature, which is not really amenable for many of these viruses. So what happens is, even if there is a virus boom at some point inside the bat, once they start flying, their body temperature increases, which in turn kind of reduces the amount of virus in the system. So they're able to tolerate all of these different types of viruses, right? And with globalization, humans are increasingly coming in contact, deforestation, all of which you would have learned in like 10th grade social studies, for example. There is this deforestation, so on and so forth. So there is a lot more interaction with bats in the wild. So, naturally there is more spillover these days.
That does make sense. One of the other puzzling questions that people have around the virus is why does it affect different people in different manner? Is this aspect of varied impact depending on the
Mhm.
host, or is it, what is that? Why are some people symptomatic and why are some people asymptomatic? What are the factors at play there uh, that gives out this
Right.
different reactions from different people infected with the virus?
Yep. So mostly, so this is dictated by the host, in terms of the severity of clinical illness, not really from the virus point of view, because, this virus is highly transmissible, right? And its ultimate goal is contagion, correct? And this is like the Virat Kohli of viruses. So it doesn't need to really change anything, to be honest, right? So all of these different clinical manifestations comes from the perspective of the host. You have certain populations which are more vulnerable, right? For example, older people above the age of 60 or 70. And age is in general a proxy for your health and also a proxy for your immune system, right? So the faster your immune system is able to control the virus, the lesser the symptoms will be, right? And again, people with comorbidities such as diabetes or cardiovascular diseases or people who smoke generally tend to have weakened immune systems. And even people who are immune compromised, those who have HIV/AIDS or are in chemotherapy, these people have lower or weaker immune systems, so they're not able to mount as much of an immune response, which leads to worse outcomes. And outside of this, another feature is that there is uncontrolled immune activation, which leads to a term called cytokine storm, right? So to kind of give you some context, what happens is that your virus kind of initially hops on to your upper respiratory tract. Okay, so if you're a healthy individual and if you have a good immune system, your immune system is able to immediately detect it. So your immune system is again kind of split into two different arms. You have the innate system and then you have the adaptive system. The innate system is very quick in response, but it is very general. It's not very specific and not as robust as the other arm, which is called the adaptive immune response, which are your B and T cells, your white blood cells. Um, and this adaptive immune response is more specific, more potent, but takes time to kick in, right? Because your body needs to kind of identify what the virus is and eventually mount a very specific response to it. So what happens is when the virus initially hops on to your upper respiratory tract, your innate immune response kicks in, right? So it kicks in, and if you have a healthy immune system, it's able to immediately clear the infection. And it releases molecules called cytokines. So these cytokines have multiple functions. There are different type of cytokines that exist, and each of these has different functions. And it's just a huge repertoire of cytokines that exist. And in moderation, it is very helpful, right? But what happens is if your immune system is not able to detect it and control it, the virus gradually moves from the upper respiratory tract, from your trachea, upper trachea to the lungs. And that's when it starts getting more serious. And then in response, the air sacs in your lungs starts releasing so much fluid, which leads to, breathlessness and eventual, need of ventilator and ARDS, which is acute respiratory distress syndrome. And outside of this, what happens is sometimes your immune system will try to overcompensate and go into overdrive, which means that in order to get rid of the virus at any cost, it releases so much cytokine, right? So cytokines in moderation will kill the virus infected cells. But what happens is that if there is a lot of localized cytokines, the immune cells will also start killing the bystander cells which are not infected, and cause a lot of inflammation and damage to the lung, right? Which is what causes your acute respiratory distress syndrome. So a lot of this, the difference in the symptoms comes down to the kind of the status of your immune system, right? And another important thing that I want to highlight is that children. So, for example, for flu, the vulnerable populations are young children and older people, right? So you would think that there should be the same case for COVID, but that's not the case. Older people are vulnerable, but children, for some reason, are not as vulnerable as they are to the flu. And scientists are speculating. They still haven't found out why that's the case. One particular reason is that children and healthy individuals in general have more or a higher density of human ACE2 than older people. This seems counterintuitive, right? You would think that if you have more human ACE2, it would, exactly, right? But that's not the case. What happens is, human ACE2, it is there for a reason in the host, right? It is not there for the virus to use. So humans have a function for it, and it plays an important role in immune modulation, right? So scientists believe that if you have more human ACE2, you have a more balanced immune response or a dampened immune response, and there is no uncontrolled cytokine storm. So that is a potential reason why maybe children have less severe symptoms as well as healthy adults. So these are a few reasons why there are just a myriad of different symptoms out there.
So on the same lines, how much of a correlation is there between the blood type and the risk of a person getting infected? I read a couple of reports where people of a particular blood type, they seem to be more at risk of getting infected.
Yeah, so there was I think the study that you were referring to was actually a very neat study that was done in Italy and Spain, where they, I think, recruited I think it was close to 2,000 patients. I'm not entirely sure. So the idea of that study was to compare the genomes, right, to see if there are any differences between people who had symptomatic disease, who had severe disease, and those who had mild disease. All right. And of course, the media will only focus on things that are that make more sense to them, which was blood type. But there were other conclusions that also came from that study, right? There were, I think, three distinct conclusions. One was the blood type, obviously, where they showed that people who had blood type A had a 50% greater risk of severe disease, versus people who had blood type O had a 50% lower risk of getting severe disease. Again, this is not whether you will get infected or not, but this is more down to how severe the disease will be, right? Whether you have mild symptoms or if you have severe symptoms. So the other um, kind of two other places where they found mutations or differences between the genome of these two groups is that one region was in uh, 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 a protein that is involved. It's called a transporter protein, and it is shown to have interact with human ACE2 in some form, right? This is another region that they found a difference in. Again, they don't know how this really affects disease outcome. And the third kind of region that they looked at was there was a mutation in one of these cytokines. So there is a cytokine called interleukin 6 or IL-6, which is a pro-inflammatory cytokine. So this cytokine will promote inflammation, right? So this in moderation is very important for viral control or any pathogen control. But again, maybe the scientists speculate that mutations in this gene could lead to, excess production of this IL-6, which could, worsen the disease outcome. So these were kind of the main talking points of this paper. And, blood type correlation was also, again, one particular finding. And again, more studies needs to be done to definitively say that this blood type would really cause worse outcomes.
Got it. So this is one of the cases where correlation doesn't really mean causation.
Causation, yep, exactly.
So that's a kind of a relief actually, because the headlines that were being thrown around is A positive is one of the blood types that is very vulnerable, and I happen to be A positive. So personally, it's really worried.
No, in general, so that's the thing, right? You need to rank the outcomes of certain things, right? So you are a young and healthy individual, so that would precede any other risk factor that you have, right? So if you're a young, healthy individual, the chances of you having a severe infection is very low, right? Irrespective of the blood type that you would have, right? At this point, that's what it seems like, right? But for people, I don't want to raise any alarms, but older people or people with weakened immune systems with blood type A, maybe you could have worse outcomes, but we don't know again. There's just, so something that needs to be put out there is the fact that this virus is only 6 months old, right? And the amount of information that we know about this virus already, thanks to so many scientists around the world, is just amazing, right? We've been studying HIV for the past 30 odd years, flu for the past 100 odd years, and we don't have a good vaccine for that yet, right? But the way in which we are progressing for a vaccine now for SARS CoV-2 is just unprecedented. So the amount of work that's going into it is just amazing. But at the same time, you should take it with a grain of salt, because some of these studies are being put in preprint journals which have not been peer reviewed yet. So whenever we publish academic papers, they need to go through a peer review process where experts in the field will review your paper, make sure that it meets scientific standards and rigor, and only then will it be published, right? But some of these reports that are coming out have been are being put up in these non-peer reviewed journals, right? So they have not been critically analyzed by experts in the field. But then, the media is always out for news, so they're going to jump onto this, right? But as a public, you can't really blame the media. They are out for news. But as public, they should, take it with a grain of salt and see if, I know it's tough for the public to do that, even for, me being in the field, it's very tough to keep abreast of all of these new findings that are, coming up, right? So you should take it with a grain of salt and make sure that this has been published in a journal that's, that goes through a peer review process.
So I want to get your thoughts on testing. I want to understand what goes on behind the scenes when a person takes a COVID-19 test, and why does it take so long to get the results, and why is it so hard to procure these testing kits?
Okay, so there are two types of tests that you can get for COVID-19. Okay, one is to test if you have the virus right now. Okay, and another test is to see if you've had the virus at some point. Okay, so there are two clear distinctions. Okay, so the first part, which is to show that you actually have an active infection, is potentially when you have symptomatic patients who go in and then you get either a saliva or you get a nasal swab. And then what happens is that the kind of process that goes through is that they will kind of dip it in a liquid. And then what they will do, or technicians in general, will harvest RNA. So this SARS CoV-2 is an RNA virus. Okay. So the genetic material is in the form of RNA versus, humans are DNA. And you also have other DNA viruses. But so what you do is that take this fluid and then you extract the RNA from this fluid, and then you perform something called reverse transcription, where you convert this RNA into DNA, like how HIV would do it. And then using this DNA as a template, you will do a technique called polymerase chain reaction, or PCR, which is kind of a staple in the life science community. They will do this PCR to see if you have um, pieces of a viral RNA. Okay, so this is the process that happens. So as you can see, there are multiple steps involved here, right? And RNA is very fragile, to be honest, right? So there needs to be a cold chain for it. And to do each of these extractions, right? um, Extracting RNA is not that big of a limiting factor, but then converting this RNA to cDNA, you need a very specialized machinery called a reverse transcription machinery, right? And right now we kind of derive it synthetically based off of certain retroviruses like HIV, not exactly HIV, but other retroviruses. So these things, it's very difficult to scale this particular reagent for mass testing, right? So that is a big limiting factor, and that is why it is nearly impossible to have billions of tests, right? Even right now we've 6 months into this pandemic and we don't have enough tests, because that is a huge limiting factor, right? So this is one type of test that they do. The other one is antibody testing, where this will inform whether you've had the virus at some point before, right? So what they do is take blood from you, harvest serum, and then see if you have SARS-CoV-2 specific antibodies in the serum. Okay, this is a straightforward test. Okay, this can be done quickly, something like a pinprick. Get the blood and put it on a membrane, and you'll know if you have it or not. Okay, but again, the problems with those is that right now you have it. I see more and more companies coming out with that. But the problem with that is that you need to validate it. Okay, so you need to make sure that it detects all positives and it negates all negatives, right? You don't want a high false positive rate, right? That can be very detrimental. So the time it takes to validate these tests will be long, because you don't want any false tests out there, right? So that is kind of the limiting factor. So I think going forward you'll have more and more of these antibody tests, but antibody tests will not tell you if you have a current infection. Okay, because antibodies takes time to develop, so you'll have good detectable antibody titers like 2 weeks after you have the actual infection, right? By the time you could have cleared the infection, right, or you have no symptoms at that point, right? So these are two clear distinctions. And all of these reagents in general, when we do it in our lab, these kits come from different parts of the world. They're manufactured in different parts of the world, and then this company kind of puts it together, right? So you need all of these different manufacturing units working full time overdrive to come up with tests, millions of tests, right? So that is a huge problem. And countries like China are like the manufacturing hub of the world, and they were shut down for a long time, right? Which puts a strain on the supply chain. So in terms of logistics and manufacturing, there was a huge problem. There wasn't a problem in terms of the science aspect of it. Of course, validation. You can validate the PCR test quickly, which they did do, but I think the limiting factor is really the unavailability of the reagents to scale it up for mass testing.
That makes sense. So, on the subject of vaccines, I have this particular question, and we can
Uh-huh.
choose to not comment as well. So a few weeks back, Dr. Anthony Fauci. So
Mm-hmm.
give context for folks listening. He's the director of
Mm-hmm.
the National Institute of Allergy and Infectious Diseases.
Uh-huh.
And he's one of the top infectious disease experts on the Trump's administration task
Uh-huh.
force for coronavirus. So
Right.
the gist of it is he basically gave a timeline of fewer than 18 months to produce a vaccine. So
Uh-huh.
that outrageously ambitious. The reason I'm
Um,
asking is,
so
so I got to read this very interesting, insightful, eye-opening New York Times piece uh, that
uh-huh.
addresses the practical challenges of developing and shipping vaccines and what a typical time
Uh-huh.
frame might look like. So, based on
Uh-huh.
what I read there, more accurate prediction is somewhere around 2036, if I'm not mistaken.
Uh-huh.
So, what is your thoughts on this whole vaccine in 18 months? Uh, is that something that we should take with a grain of salt?
No, actually, just to kind of to start off, Dr. Fauci is like the superstar of infectious diseases. He is an expert, and all of us look up to him. Again, he is in a very precarious position, trying to kind of manage this, to be blunt, inefficient administration, as well as at the same time help the public, right? So I think this 12 to 8 months was kind of a trade-off to please both ends of it. But in terms of vaccine development, right, the fastest a vaccine has been approved was 5 years. Okay,
Oh,
and this was back in the 60s or 70s. I'm not really sure. So that was the fastest vaccine has ever been um, uh, approved, right? Because there are, like I said before, there are a lot of these phases, and FDA sets really high standards for vaccine safety and efficacy, right? And for you to not cut corners and go through each of these stages, it can take like 15 years, right? So initially when we started working on vaccines for animals and humans, my boss will always tell me that vaccine development for animals will take like 5 years, right? Because the standards are a little bit lower, but then vaccine development for humans will take like 10 to 15 years. Okay. And my boss has developed vaccines for TB, for other food animal pathogens. And, some of them have not been licensed yet because it takes a long time to show that it works and it meets the safety standards, right? So right from the start of preclinical testing till when it is approved, it can take about 10 to 15 years, right? And then the next big limitation is manufacturing, right? Like I talked about the tests, some of these new technologies that are there in phase 3 clinical testing are newer technologies, right? Those that have not been used previously for mass vaccinations. So how are they going to manufacture that at a large scale? From my perspective, I don't really know, but I'm pretty sure they're working on it at the moment. But it's just, but again, that scientists are moving at an unprecedented rate, and most of this preclinical testing has already been done for MERS and SARS-1, and because of these pharma companies, big budget, they were able to also do their preclinical testing for SARS-2 within 2 months. They developed the vaccine within a month, tested it in different animal models with 2 to 3 months, and then immediately started clinical testing, right? So the worry now is that we really don't know what's going on with respect to the clinical trials. The hope is that in an effort to kind of come up with quick vaccines, the hope is that they do not cut corners, and they do not compromise on the safety of the vaccine in an effort to come up with a quick vaccine. So 12 to 18 months is highly ambitious. In my mind, I can see a vaccine that can be given to everyone possibly by the end of 2021 or start of 2022. And that in itself is very ambitious. But then all of these vaccines that are in, clinical trials right now are very promising. So we know that they work, right? Again, the thing now is whether they are safe, number one, And number two is if they can be produced at a large scale, right? We need 7 billion doses for the whole world to be vaccinated. Will we ever reach that stage? We don't know, right? And many of these vaccine companies are based in Europe and in US. So how are countries like India, Pakistan, or Bangladesh ever going to get their vaccines, right? So these are the questions that need to be answered, and the more practical limitations outside of science.
So one more question that I probably want to get your views on without offending you in a sense is what is it that makes preparing for a pandemic so excruciatingly tough? Because at least from my layman's understanding, given our access to resources, our tech advancements, we talk about AI and whatnot, shouldn't we have had a head start? When you look at that now famous Bill Gates TED Talk, he basically predicted this like he had a time machine. You can't help but think we could have done something different at some point here.
Right. So again, the biggest problem is funding, right? So scientists have been harping about this for a long, long time. So this is kind of an integral part of Virology 101 that I took. My instructor at that point said, there is going to be a pandemic in the future. We just don't know what it's going to be, right? But we do have the ability to know what it could be, right? And that again comes down to surveillance, right? And all of these new emerging infections are zoonotic infections. They are coming from animals, right? Why can't scientists, go about surveil? I know it's a huge undertaking, right? Go about surveilling bats and other rodents to see what are the viruses that are going on and develop vaccines and drugs preemptively, right? For SARS-2, take SARS-2 for example, right? We've had SARS-1, right? And many groups were working on SARS-1 and it eventually died off, right? And no one really pushed after that. And scientists were clamoring for funding. No scientists got money to pursue SARS-1 because the government and other these big pharma companies thought there was no profit and no point of doing because it eventually died off. But if we had developed drugs for SARS-1, the biology of SARS-1 and SARS-2 is similar enough that you can translate drugs that you develop for SARS-1 for SARS-2, and we could have saved so many lives. And the same is true for vaccines. If we had a vaccine for some of the more conserved regions of SARS-1, we could have used that for SARS-2. That would have had some efficacy and again saved so many lives, right? The problem is that in many parts of the world, vaccine development comes down to the hands of the private sector, right? These large pharma companies don't want to spend millions and dollars of, on vaccines that could not be used at all, right? Say you develop vaccines preemptively, and what if there is no pandemic in the future for those vaccines? So, and life science research is very expensive. So pharma companies are not going to invest millions of dollars into developing a vaccine that they might never use, and there is no monetary sense to do it, right? So the shift must come from, from above, where federal government agencies should take it on themselves to start developing vaccines preemptively and keep a stockpile, right? Pump research into surveillance, pump research into developing vaccines for these animal viruses. And then if there is a pandemic in the future, we can immediately start scaling up and using that, right? So that is how we can be ready, right? It is very tough to stop these spillover events because, the world is becoming smaller and smaller. It is very, tough to kind of prevent these events from happening, but we can be prepared. And how is that? By kind of redirecting funds to surveillance and developing drugs and vaccines for many of these viruses that are circulating out in the environment.
Yeah, that makes a lot of sense. And we are fortunate enough that someone like a Bill Gates or a Warren Buffett, people who have real influence over things like this, are actually raising their voices to invest in such initiatives preemptively, like you mentioned. And the worst part is probably you have these internet trolls who still take a dig at Bill Gates. He's trying to rule the world.
Yep.
I personally admire him a lot, and
Uh-huh.
I've been following him for quite a while, and he
Mhm.
does look like he has genuine interest at heart.
Yep. Something from our perspective is that Bill and Melinda Gates Foundation funds a lot of research focused on diseases in tropical areas, right? Um, con- um,
Mhm.
Conditions like malaria, diseases like malaria, and then TB, and then so many other diseases in Africa. Many groups in the US are studying these diseases, and all of them are funded by the Bill and Melinda Gates Foundation, right? They are very generous, and Bill Gates needs to be, appreciated for his philanthropic efforts, definitely. And I mean, there are always going to be people on the other end of the spectrum, but that's how the world is. So you can't do much about it.
That's very true, man. So I have a few
Uh huh.
people who came up with a lot of interesting questions that they wanted your opinion on.
Okay.
So I want to jump into the Q&A now. We had a couple on the transmission, and there is probably some of which that we have already covered in the course of the conversation. One of the questions is, I see a trend where the incubation period for people affected has risen from 14 days to 21 days. Is there a case for the virus getting more comfortable and stronger within the human body? That's actually very interesting.
Yep. So I haven't come across any studies which have specifically looked at that. But then again, there's just so many of these papers that are coming out these days. It's tough for me to keep abreast. But uh, in terms of the populations that are getting infected, maybe there is a shift from older people to younger people that could potentially explain why there is a longer incubation period. Because the virus is finding it tough for to kind of establish an infection and before symptoms start to appear. That could be one reason. The other reason is not necessarily reason, but at the genetic level, right? There are um, very few mutations that have a phenotype per se. So there are few mutations that have come up in some of the circulating strains, but that does not really translate into a difference in how the virus transmits or replicates in humans yet. Because again, this is, like I said, is the Virat Kohli of viruses, right? So it doesn't really need to change anything, right? Unless there is a bottleneck. Say you come up with some silver bullet drug in the future, and there is a bottleneck there, and maybe at that point, mutations could lead to drug-resistant viruses, right? So as of now, in my understanding and knowledge, there has been no development of any of these new viruses or new mutations or anything, or better adaptation, right? I think to begin with, these viruses are very well adapted to humans.
Okay, so I'm going to move on to the next one here at this point. This
Uh-huh.
one is on the transmission dynamics of the virus.
Uh-huh.
There were actually a couple of questions on the same subject. So one was about why the symptoms are not uniform or distinct,
Uh-huh.
that we
Right.
covered at depth in one of
Yep.
the earlier parts of the conversation. So the
Yep.
other question we have here is, because we have a lot of debate on whether to order food and the safety of it. So does
Uh-huh.
this virus spread through food? Say somehow it landed on my
Um,
food and I ate it. What are the chances that
mhm.
I'll be infected?
The chances are very low. Again, there are multiple things that you need to take into consideration here. Is that this virus um, is very sensitive. These, getting into the technical side of it, these viruses are envelope viruses, so in general they're more sensitive. But like I said, these SARS-CoV-2 infects a particular type of cells, which is present only in the upper respiratory tract. Right, the food that you ingest does not go through the upper respiratory tract. Right, so the chances that um, you would really get infected with SARS-CoV-2 on food is very, very negligible. Right, but people ordering food from outside, what I would suggest is to kind of clean the surfaces of the packaging that you get, but don't really look to sanitize any of the food per se. If you just
Obviously.
clean fresh fruits and vegetables, that's more than enough. But usually they are just there on the surfaces, So just the containers or packets, if you sanitize it well enough, it's fine. There is literally no way it's going to transmit via food. And even one of the common symptoms that people have been noticing is diarrhea. So which means that the um, virus is able to establish infection in the gut mucosa, but then again, it needs to start the infection from the upper respiratory tract, right? It can't directly go to the gut and initiate an infection, right? So it is a gradual process. So you can't really, the long story short, you can't really get infected from food, eating food.
Very relieving.
Yeah.
So I have one on immunity here.
Uh-huh.
As someone who has no choice but to go out on the field for work,
Right.
and possibly even interacting with people who have tested positive eventually, I have taken the test 4 times already, and it has all come back negative. Is it possible that I might have encountered the virus without actually contracting it? So,
Is this
also as a follow up on that, if a test comes back negative, how much can I actually do it? So the reason I am very specific about this is I have old folks at home, and I'm constantly afraid I might have the virus without getting infected, but I'll still spread it to them. So is that clear, or do you want me to go through the question once more?
No, I think that's clear. So I think a distinction for the kind of the listener to clarify is what type of testing they got, right? So like I explained before, there are two different types of tests. You have your antibody testing, and then you have your PCR test where they look at active infection, right? So if you did the PCR test and it came back negative, it means that, and 4 tests, right? In general, sensitivity is a measure that is used to evaluate the performance of a test, right? How sensitive is it, and how quickly can it pick up the right positives, right? But if you've done it 4 times in succession, the kind of the sensitivity rate increases from 99% to 99.99%, right? So you can be sure that you have not had the virus at the 4 times that you have tested, right? The other thing that you could possibly do is to get an antibody test, right? So this tells you whether you had the virus at some point. Okay, so there are 2 kind of clear distinctions. The chances that you don't have the virus is pretty high since you had 4 negative tests. But if you are really curious and want to know if you've had the virus at some point before, then I would suggest that you do an antibody test.
I'm sorry to interrupt, but do you have any idea on the kind of tests that people generally take? Because in India, it's just we have government institutes that mostly just if we have symptoms, we just go there and take a test. So there is probably only one kind of testing that's done across the country.
Yep, that's a very good point. Yeah, so I think that must be the PCR test, which tells you whether you have the infection at that point or not. Okay, these antibody tests are slowly coming up in the US, and I know that there are some vendors who could probably eventually market it for the public, so the public can directly buy it from pharmacies in India and do the test themselves. It's like your sugar test or something where you just prick your finger and then you get the blood and you see if you have antibodies or not. If it is positive, it means that you had the virus at some point. But if it's negative, it means that there is a high chance that you've not had the virus before.
So the follow-up question this person had was, is there a chance that they might have the virus without getting infected? As in, does it even make sense? That's something that I've been confused as well. Is there a case where you carry the virus but you're not infected by the virus?
Yeah, there is a possibility if there is very low level replication of the virus. And in general, studies have shown that 48 hours before symptoms kick in is when there is a high viral replication, right? So there is lots of virus everywhere, which is eventually picked up by the immune system, and then you start having these symptoms. right? So at that point, like 48 hours before symptom onset, you could start shedding virus. And again, this is not being asymptomatic. This is pre-symptomatic, right? You're going to get symptoms at some point. It's just that you start shedding the virus. And again, this asymptomatic or mild symptoms are very subjective, right? People can have as low as a sniffle, as small as sniffle, which they could, consider to be just allergies, but it was actually COVID. So it's just a total range of different symptoms that are out there. So it's very tough to gauge it at this point. But scientifically, yes, it is definitely possible for the virus to kind of replicate without any symptoms if there is low level replication, which could potentially still facilitate transmission. That I do not know, but in my mind, I think it could.
So it basically means, to sum up what you said, there is a very good chance that I might carry the virus, but I have a very good immune system, but I might still end up spreading it to people near me. Yeah.
Yep.
Wow, that's actually scary, man. So,
yep, that is, I think, one of the biggest drivers of transmission for SARS CoV-2. This pre-symptomatic or asymptomatic spread.
Okay. So I have this very generic question, but something that we have all been pondering here back in India is: are masks genuinely useful?
Or what?
Masks.
Sorry, I didn't catch. Yes, definitely. So masks will prevent, say you have COVID or you have symptoms, it will prevent others from contracting the disease, right? So as a community, just like taking vaccines and gaining herd immunity at some point, it's important that all individuals in the community wear masks. So, so that it might not prevent you from getting infection, but it does, prevent you from transmitting the virus. So I think the chances of you spreading the virus reduces from 70% to 1%, even if you wear a normal surgical mask and not even your N95s, right? And with N95s, the chances go down even lower, So, and with N95s, you can also prevent contraction, right? So with normal surgical masks, you can, prevent kind of giving the disease to another person or transmitting the virus. But with N95s, you can also prevent contraction, right? Because they prevent like 95% of, I think, small particles below a range. So they are pretty effective. But again, there are ways in which you should wear an N95. If you do it properly, you can prevent, contracting the virus.
I came across this very interesting article. There were a couple of stylists in Missouri. So working in a salon, and apparently they got tested positive, but they didn't know that. They went about just basically styling people in their salons. They interacted with about 140, 150 odd customers before they eventually got to know that they had the virus. But luckily, none of the people that were in contact with these two stylists, they got the virus because they were wearing masks.
Masks, yeah, definitely, yeah. So I think masks play a huge role, and I think US was wrong initially to have not suggested the use of masks, but I guess that kind of suggestion stemmed from the fact that there was a shortage of masks and they needed it for the healthcare setup initially, before the supply kind of met the demand. But yeah, I mean, masks are very helpful, and I highly recommend that people wear masks all the time when they go outside.
So I have the last question here. What are your thoughts on Ayurvedic medicine, such as Patanjali, who are advertising their drugs for the coronavirus?
Okay. So the thing with Ayurveda is that, there are a lot of these medications or therapies that have been studied in a scientific manner and have been proven to be effective, right? But a large part of it has not been scientifically proven, right? And as a PhD student, you're trained to view everything critically. And the point here is that have they really done any of these clinical trials to show that this would work in humans? No. And for example, look at hydroxychloroquine, look at remdesivir. All of these two drugs were touted to be the kind of the drugs for COVID, but none of them worked when they tested humans, and then they closed it completely. And then now they're looking at other opportunities. Can you do the same thing with this Ayurvedic medicine that's come out? There are some benefits to for Ayurveda, for other diseases or conditions, but it has not been tested for coronavirus or for COVID. And I don't think people should take it, and they will be under this false impression that they will be protected, and they're going to go about contracting the virus and then transmitting the virus to everyone, because we've not really tested it. The chances of it working, yeah, potentially it could work. As a scientist, you are led to believe there are two sides to a coin, so there are chances that it could work, but I'm going to base my answer on evidence, right? Unless I see data to show that it works, I'm not really going to trust that. So that's my standing on this whole Ayurveda kind of controversy that's floating around.
So that's it from the aspect of the crowdsourced questions that I have here. And I do
Awesome.
have a couple of questions before we wrap up for the day.
Okay.
I know throughout the episode we have it has taken a rather pessimistic tone toward the end, at least as we progress.
Right.
I mean, merely because of the situation we find ourselves in. So you look at the vaccine's not available, you have folks dying. But so is there anything, any kinds of progress that we have made so far? Any positive note to close with? I'm particularly keen on a couple of names that got thrown around in the recent weeks. And one was the remdesivir, and the other was the mRNA. So you mentioned these names in the course of the conversation as well. So is there anything interesting there? Any glimmer of hope at this point?
Yep. So kind of there are two different interventions that you can take for COVID. Okay, one is you have vaccines, and the other is that you have drugs. Right? Vaccines are something that is very important in eventually eradicating the virus, like we did for smallpox, for example. Is that it prevents disease in itself completely, right? And then you have drugs, which is post-exposure, right? So once you get infected and you have symptoms, you will treat the symptoms with this drug. It can either be an antiviral, which will specifically target the virus, or it will be something that is immune modulatory, something that will dampen the immune response so that you don't have a bad inflammatory response, right? So these terms, these mRNA and DNA are all vaccines, right? You have certain companies such as Moderna, they're working on these mRNA vaccines, and the preliminary data is very promising. Even if you look at the preclinical data on mice and hamsters, all of the mice and all of the hamsters that were vaccinated have got something called a neutralizing antibody response, which is a proxy for good protection, okay? And then when they translated it to humans, I think more than 80% of individuals who got the vaccine had neutralizing antibody responses, which is very good. And I think this is true of other vaccines that are currently in clinical trials. So the data is very promising. So the chances that it will work in the field is very, very high, right? But the question now is, when is it that we'll get it? So that is a big question there, right? So vaccines we know will work, but it takes a time to produce, right? And then now moving on to drugs. First, there was a lot of media attention around hydroxychloroquine and then remdesivir. Hydroxychloroquine, again, the mechanism of action is unknown. We don't know whether it directly targets the virus or if it is immune modulatory. And remdesivir is also, it's proprietary. It's from Gilead, and we really don't know how it works, right? But then both of this was tested, and they did not seem to improve outcome of patients, right? So, for example, I think for remdesivir, the difference was like 11% of people who had COVID had severe outcomes, right? But with remdesivir, the kind of percentage um, reduced from 11% to around 9%, right? So this is not a huge difference in terms of outcomes. So both of these drugs, and for hydroxychloroquine, they actually found out that people who were administered hydroxychloroquine had worse outcomes
Wow.
than people who did not get hydroxychloroquine, right? So because of these substandard results, both of these drugs have been shelved. Okay, but then there is this new drug called, I think it was dexamethasone or methadone. I can't really get the correct generic name out, but this is, I think, is a commonly used steroid. And I was just looking through the data. I think it was a couple of days ago was when it came out, and they've shown that it specifically targets one group of people. So people, people with severe infections need ventilator use, and I think half of the people who eventually get onto the ventilator do not make it, right? And these are the stats within the country, in the US at least. This steroid has been shown to improve outcomes who have eventually gotten onto the ventilator, right? So that is promising. So this could potentially reduce the mortality rate. And this, again, is not your silver bullet, but it will help reducing the deaths associated with COVID. And this is ubiquitously available everywhere in the world, right? This is a commonly used steroid. Right? And some of the other positives that you can probably look into is the fact that at least in India in general, the mortality rate, especially for example, Tamil Nadu, is very low, because again, I feel like they're doing a good job within their limits. And the other thing is that in India in general is a very young population, right? So the severity of disease is going to be lower. So proportionally, the mortality associated with that is also going to be lower, right? So these are some of the positives that I can think of, and the progresses that we've made.
Awesome. So I just have one last question.
Uh-huh.
When you look at countries like US, India, and
Uh-huh.
the rate at which the cases are growing every day,
Uh-huh.
is trying to flatten the curve even relevant at this point? Or is it just a matter of waiting out for a vaccine or counting on herd immunity?
No, I mean, definitely flattening the curve is very important here, because the stress is on the public health system, right? Your healthcare infrastructure, right? Say, COVID is, there are going to be 1,000 cases of SARS CoV-2 infections in an area. You can have that either over a week or over a month, right? And the stress on the healthcare system is going to be different in both cases, right? So it is very, very important to flatten the curve, especially for countries like India, where I think all of your listeners will also agree that the healthcare infrastructure is not up to the level of some of these Western countries, right? So there are a lesser number of ICU beds per capita. So you need to flatten the curve as much as possible. And I felt like India kind of locked down pretty early, which was good. Although you can, comment on the ways in which some of these were implemented, but still, the fact that the lockdown was implemented early was good. And you can see that, right now we have a lot of cases, but this has happened over 3-4 months, right? So there's been a gradual increase, but so the healthcare system has been able to cope up to an extent, right? Versus, say, for example, in the US, where right now there are 40,000 cases yesterday in a single day, right? So that is something that India cannot afford. So you need to flatten the curve. And how can you flatten the curve is by, isolating yourself if you can. If you can work from home, please do that. Limit the times that you go outside of your house. And then wear masks all the time that you go outside. Maintain social distancing, 6 feet and 12 feet if possible. So if you do all of that, you can flatten the curve as a community and help the healthcare system. So that's what we are doing right now. And we need to be doing this until we have a vaccine or a drug, that is shown to help people.
Mm-hmm. Wow. Thank you, Shaswath. It's probably one of the most enlightening and insightful episodes that I have done personally. And I know how busy you are and the kind of job you're doing, and for you to make up as much time as you did, especially on such short notice, big thanks for that. And I hope people
Yep,
listening
definitely. I think, yeah,
something.
the pleasure's all mine, Krishna. I mean, to give you some, I've talked about this before as well, but I'm a big fan of podcasts myself. So it's just, cool that I'm coming in to a podcast as a guest myself. So there is some personal motivation from my end too, apart from the greater good and the fact that, I also want to be able to communicate science and also inform the public of the right things to do, right? And answer some of the questions as well. So yeah, I'm happy that you contacted me and we were able to do this.
Wish you all the best in your fight against the virus.
Yep, thank you, Krishna, and keep doing this.